and hopefully you'll get some time to rest, and study a lot, right? Okay, so I just uploaded the PowerPoint for today, and we are going to start expanding our knowledge and going into cellular immunity. And this is a lot of fun, at least for me. Okay, I really like this topic because it's so challenging and it's kind of like a puzzle that you have to put together and see where the pieces are going, and the pieces behave differently if you combine them in a different ways. Anyway, you're gonna see what I mean, right? So here, starting with the cellular immunity. Okay, before we go into cellular immunity, I just wanted to say a few words about those slides that we had left from the last lecture, right? So last time we were talking about the secondary lymphoid organs. So yes, you have to know about their structures and their similarities, and the cells where they are hanging around, and how they're interacting, right? So we talk about the lymph nodes, and then we were finishing to talk about the spleen, and then I stop at this slide, right? So functions here of the spleen. Remember, the spleen is going to filter your blood, right? Can you survive without the spleen? Yes, some people have their spleens removed. When would you have the spleen removed? What would be a situation that you should take the spleen out? Really? The spleen has a lot of the secondary lymphoid cells that yes, when it ruptures, right? So the spleen, remember, you have that open type of circulation. The arteries are not directly connected to the veins. So when the arteries bring the blood to the spleen, to the splenic artery, right? When the blood comes inside of the spleen, it spills out and kind of like gets dumped over there. So the only thing that is preventing your spleen from killing you from internal bleeding, right? The only thing that is preventing the spleen from doing that is the outer shell, is the capsule of the spleen. Now, how tough is that capsule? It's tough enough that if you fall on the floor, it's not going to break, right? But if you're playing some type of contact sports and some other crazy sports that I never understand the reason for the sports in the first place, like American football, you know what I mean? They put their helmets, which I think are kind of weapons, and they put their helmets and they aim at somebody else and then kind of buck their head in their stomach or right into the spleen. You know what I'm talking about? Like physical sports. Or there you have the wrestling sports, high contact sports, right? That one I understand even less. In this little circle, and you go like monkeys and you kind of like grab each other. And anyway, those kind of sports, high contact and possible injury, a lot of times the spleen can get damaged. If you break the capsule of the spleen, blood is going to spill out of there, and if you don't stop the bleeding, you're going to die. You're going to die of internal bleeding. Okay, so this one you get your spleen removed. Now, if you get your spleen removed, well, what are the functions of the spleen in the first place? Oh, here they are. They are going to remove all the red blood cells or damaged red blood cells. They're going to help to fight viruses that got into your bloodstream, okay? And they are going to get rid of opsonized cells as well as red blood cells, opsonized pathogens, okay? Now, if you take your spleen out, who's going to take care of that? Mainly your liver. Your liver is going to take care of getting rid of all the red blood cells, okay? And most likely, some degrading phagocytes are going to take care of breaking down some other opsonized cells. But then you have a problem that you don't really have all the immune functions that you have in the spleen. You don't have that in the liver, okay? So people who have their spleens removed, they tend to have more infections than other people who have their spleen intact, okay? So now when you look at the circulation in the spleen and you see that, the blood gets there and spills out, so you're going to see, well, how is it that the blood is going to get to the venous side, right? So this is what you have to keep in mind. The blood spills out, it gets filtered, macrophages are going to eat up the pathogens, etc. And then the good red blood cells are going to get back into the circulation. How? Well, they are flexible and they're not tagged, they're not bulky, so they can squeeze through little slits that are present at the venous side of those blood vessels inside the spleen, okay? So if you have any other tissue in the body, any other organ, you have the arteries that are connected to the veins at the capillary beds. So here there's no capillary bed. There are arteries spilling out the blood, and then the veins, they're going to collect the blood that is good to be recycled. And here is how the end of those little veins look like. Okay, so it's not like there's a hole here. So this is basically like covered. Okay, so this is a bit misleading. This part here is kind of like covered like that. So you don't really have. So here's a continuation of cells. It's not open here. Okay, the only parts here that are open are those little slits, and you call those slits, what do you call them? Everybody's awake? Right there, interendothelial slits. Okay, so they are here in the endothelial cells, and they allow healthy red blood cells to get back into the circulation. Okay, now if the red blood cell is not healthy, it's gonna be eaten by the big guy, the macrophage. If the macrophage finds some crazy swimming bacteria in the blood, it's also going to eat that. That's why it's called the big eater, right? Now, those macrophages, they are specialized in recycling iron. Okay, so they're responsible for recycling, that big molecule that carries iron, which is the hemoglobin, and also to recycle iron. Okay, so this is why when you do this recycling of old red blood cells that are broken or damaged, when that happens in the spleen, you don't get hemoglobin cytotoxicity. You don't get toxicity of iron in your system. Okay, because those cells are specialized in cleaning up those old and damaged cells. So here is that summary slide telling you what is important to know about the spleen. And remember that I also added a video that I really, really like. The guy goes, step by step on all the parts of the spleen and all the functions, the white pulp, the red pulp, the pulp. So please watch that video, okay? Because I think he does a better job explaining about the spleen than I did. Besides lymph nodes and spleen, you also have those mucosa associated lymphoid tissues, and in general you call those mouth, right? What do? They're going to protect your mucosa, and mucosa, remember, is coming into contact with your outside world, right? So after the immunologists knew about this mucosal tissue, so they started to get more specialized, and they say, oh look, when you have your nose or your nose and the mouth, let's call that nasal associated lymphoid tissue, or NALT. And somebody else said, oh look, but now here is associated with the bronchus, bronchi, so let's call it bronchus associated lymphoid tissue, or BOUT. And then somebody else said, okay, now the gut one is really important, so we call that gut associated lymphoid tissue, or gout. They gotta have a sense of humor, right? So now you have mouth, naut, bout, gout, and you can do a little song with it. Okay. So within those mucosal-associated tissues, you have some specialized structures, right? So you have those things that are tonsils and adenoids. They're basically, the same thing, but they do a bit of different function, okay? like the tonsils, you can actually see the tonsils, okay? You can see your palatine tonsil. Let me see here. Where is the palatine? Right here. So if you look at the palatine tonsil, it is, if you look in the mirror, you're going to see in the back, like going towards your throat, you're going to see these structures that are on the sides of that. Oh, now I forgot this structure, the name of this little structure. What do you call it? No. Epiglottis is what blocks your airways when you swallow. You know, the sininho, you know, the mouth, the little bell is not a bell. No, no, epiglottis is in your throat. Huh? Huh? Uvula, there is it. Yeah, uvula. Uvula. Although it doesn't look like a grape to me, but anyway. So look on the sides of your throat, you're going to see your palatine tonsils. So there are a couple of them. And then if you really like you stick your tongue out and you look all the way in the back of your throat, you're going to see here your lingual tonsils right there on the back of your throat, okay? And that's also, you know, important for taste. Okay, you have papillary buds close to your tonsils. Okay, and let me see here else. Pharyngeal. Where are the pharyngeal tonsils? So those are palatine, lingual. Okay, so you cannot see the pharyngeal tonsils because they are in your throat. Okay, now when you look here at pharyngeal adenoid. Okay, so here they are interchangeable, but the adenoids, if you look at this, is like all the way between, like after your nose. Okay, you see this is an entrance for your nose and nostril here. Okay, so all the way in the back. So those adenoids, they are not really connecting with whatever you swallow. Okay, so the adenoids are mainly going to filter the air. So they're going to be trapping stuff that is coming through the air. Now the tonsils are mainly going to trap stuff that you're putting through your mouth. Okay, and it could also be the air, right? Sometimes you breathe through your mouth too. Okay. All right, so I put this slide here to differentiate the tonsils from the adenoids. So here you can, read on your own, and you're going to see basically their functions. So I put some functions here that are more important, right? Adenoids are going to be catching airborne pathogens, and then tonsils are going to be catching whatever is pathogen that comes through your mouth. Okay, so here is a little picture showing your gout. Did I talk about the M cells here before? No, I never mentioned those microfold cells. Okay, so when we get to the mucosal immunology, I'm going to mention a bit more about these M cells, but they're really, really interesting because they are on those, like sitting on those Peyer's patches. Okay, so if you look in the intestine, you're going to see that your the villi and the microvilli, they have little areas in which they have this concentration of immune cells, right? It's just like the lymph node, but it's not encapsulated. Okay, so it looks like a lymph node, but it doesn't have a capsule. This is a main difference between Peyer's patch and lymph nodes. Okay, so what happens here, like at the very top, right at the surface, close here to those microvilli, you're going to have some very specialized cells that do not have microvilli, which is actually not showing here, but those cells, they are very important at allowing samples of whatever is inside the lumen of your intestine, to be shown to the other side. And those cells are really cool because when you have friendly bacteria, those cells are important to teach your immune system not to attack the friendly bacteria. And when you have pathogens, then those cells are going to be important to bring the pathogens to give it to dendritic cells that are going to, process the pathogen or take it to the Peyer's patch and activate some B and T cells. Okay, so it's a lot of things are happening in the Peyer's patch that is very similar to what happens at the lymph node. Okay. But those microfold cells are really cool cells of Peyer's patches. Okay. Oh, so here I have more on that. So here is a microfold cell that doesn't have those microvilli. Okay. So the other cells in the intestine, they do. So these guys are kind of bald. Okay. So they help samples or antigens that are in the gut to come across without being destroyed. Okay. So they come across without being destroyed, and they are presented to immune cells, mainly to innate immune cells, like you see here, macrophage and dendritic cells. And in case they find pathogens, those cells are going to go here in other areas of the Peyer's patch. In the center here, you're going to find B cells. Close to the B cells, you're going to have the T cells. So the same type of arrangement as you find in your lymph node. So in case they find pathogens, they're going to come and activate the T cells. T cells are going to interact with the B cells, and then you're going to get activated lymphoid cells, right? Lymphatic cells here. And those lymphocytes are going to come out of the Peyer's patch, and they're going to go to the nearest lymph node. So the lymph nodes, they're responsible for surveilling the mucosal tissue. Those are called mesenteric lymph nodes. Okay, so you have seen that before. It comes out from the Peyer's patch, go to mesenteric lymph nodes, and then you have further activation and proliferation of your lymphocytes. All right, so now we get into cellular immunity. Okay, so going over here, these objectives is mainly about the T cells, right? And is mainly about adaptive immune system, but a lot of those lymphocytes are going to be part of your innate immune system, as we're going to see. Okay, so you're going to see what is happening to the T cell differentiation. What are the subtypes of T cells? And then we're going to look at some strange subtypes of T cells, right? So you have some T cells that are important for bacterial or viral infections, other ones that are important for helminths or parasites, parasitic infections, other cells that are important for inflammation and also intracellular parasites. And then you have those regulatory T cells that have to calm everybody down, right? And they all function based on what kind of message they're getting and what kind of cytokines they're exposed to, right? And then you're going to look at those cytotoxic T cells and how they are performing their killing, okay? All right, so here are supplemental videos that are very helpful, and I know there are a lot of videos. Believe me, they are very helpful, because the more you go through repetition of the topic and you hear it from different people explaining more or less the same thing, the more you are exposed to it, the better you're going to understand it. Okay, getting familiar with all this topic is not easy, it's not second nature, it's not an intuitive kind of thing. Okay, so hang in there, okay, and watch the videos, right? And after you watch the videos, what do? Well, while you're watching the videos, you should be taking your own notes, right? After you take your own notes, you should use AI to come up with some explanations of what's going on and come up with some cool STEM questions, multiple choice questions, right? And you should say something like, well, how do I apply this knowledge in a real world setting? Like how, write multiple choice questions that put this into a situation that I can actually learn something out of it. Okay, so do that and let's see what the AI answers you. Okay, be careful with AI because sometimes they give you some answers that totally don't make sense, right? When the stuff is not making sense, what do? You go back to the PowerPoint and you say, well, what was mentioned in class? It's like, if it's not going hand in hand with whatever AI is saying, what do you do then? Who do you believe? Believe me. Or you can go to my office, right? And you can ask some questions, right? You're, always welcome to go and read the book. Good luck with that. I was trying to prepare the lecture reading the book and I got more confused. So anyway, As you know, I try to make it as simple as possible, right? So let's see what you have to do here. Okay, cell-mediated immunity. Why do you need it in the first place? Cell-mediated is that cell-to-cell contact, the mano-a-mano type of fight. Why do you need it? Well, B cells are, kind of part of that cell thing. No, not really. MHC is what the T cell is going to work with. No? Well, it's actually a simple answer. You need this type of immunity because not all of the pathogens are outside of cells. Some pathogens are going to hide inside of cells, like, for example, viruses, or intracellular bacteria, right? Tuberculosis bacteria, right? So you can have some pathogens that go inside of the cells and they divide inside of the cells. And if you have just innate immune system cells going around and patrolling for whatever they can find outside of cells, they're not going to see these cells, those pathogens, because they are hiding, right? So now you have to have a system that is going to detect when you have pathogens hiding inside of the cells. So this is why you need cell-mediated type of thing. Like you're going to have cell-to-cell contact to identify if something needs to be destroyed or not. Okay? So those cells are going to be activating other cells. They're going to be working together with other cells. And the main idea of this is to defeat intracellular pathogens. Okay? Who is the main player here? The T cell. Okay? You have T helper cells that activate other cells, and you have cytotoxic T cells that directly kill other cells. Okay. All right, so here, cell-mediated immunity. You're interested really in getting rid of intracellular pathogens, right? Now, when you look at the blood, we know that the blood has like the watery thing, right? The serum, and then you have all those cells, the white blood cells, the red blood cells, right? If you spin down all the cells out of the blood and you end up with the serum, right? So serum without cells. So what do you find in the serum? Do you remember what you find in the serum? Cells. No, you don't have cells. You spin them down. Yeah, the plasma is, the serum is the plasma. What is in there? Proteins, right? You have water, you have proteins that are in solution in there. Are you going to have complement proteins in there? Okay, let's go for the vote. Who thinks yes? Okay, 1, 2, 3. Okay, go. I mean, you vote. I mean, I gotta see your hand. Don't go like this. Do you think you have complement proteins? Yes. What are the complement proteins? Part of your innate immune system. They're always floating around, and they're not cells. They're just proteins. So you're gonna have that in the serum, right? So if you get serum from somebody and you put it in somebody else, are you gonna transfer those complement proteins? Yeah. Are they going to work on somebody else? Yeah. Okay. What else are you going to have in there? Antibodies. Antibodies are proteins. So when you get the serum without any cell, you get all the antibodies in there. If you transfer this to somebody else, are they going to get the antibodies? Yeah. And this was the main method of trying to save a few people that were having very hard time with COVID. Remember? It was like a serum therapy or antibody transfusion, right? So that's a very good, when you are very much desperate and you don't have time to make your own antibodies, you can borrow some antibodies from somebody else. It works, they do work. The only issue is that proteins don't live forever. They have a certain half-life and they are going to get degraded, right? So antibodies are going to last maybe for about 3 months or so. When you're lucky, they can last a little bit longer, but they're going to die, right? So what is going to produce more antibodies? plasma cells, right? Now, if you transfer the plasma cells, are they going to produce antibodies in the new person? Okay, that's a complicated question. But let's keep that in mind. If you transfer plasma cells or T cells from one person to another, are you going to give them cell-mediated immunity? The answer is no. Because remember, your T cells were trained, the T cells you get from the blood in the periphery, they were trained to recognize something that is very specific to your body. They are trained to interact with MHC class 1, MHC class 2, and they have seen your antigens, your self-antigens, right? They have not seen antigens of anybody else. They are their self-antigens. So if you transfer T cells from one person to another, you might actually be doing some harm, okay? Because if those T cells are not destroyed right away by your own immune system, they can actually go and recognize some things that they don't see as self, which is basically everything else in the person, right? And they can start some reactions that can potentially kill the person, okay? So cell-mediated immunity cannot be transferred from one person to another, okay? Unless you are identical twins. But, you know, usually if you're identical twins, your twin is going to have the same health problems as you have. So, okay, you can't do it, right? No cell-mediated immunity transfer from one person to another. Okay. So here. Okay, do I seal that? And again, if you spin down the cells from the blood and you end up with the serum, you cannot really transfer cell-mediated immunity to a recipient with that way either. Okay. Right, so let me see what's important here. Okay. All right, so the function of this system is really to make contact with other cells and activate them or kill them. Okay, so here you have a cytotoxic T cell. So this is a CD8 positive T cell that sees a cell that is infected by seeing the antigen presented on MHC class 1, right? So now the cytotoxic T cell can interact with the infected cell and kill it. Okay. And here you see examples of helper T cells. In this case is a Th1 helper T cell that recognizes that a macrophage has eaten something and is presenting something on MHC class 2. This helper T cell goes there and totally powers up the macrophage, so the macrophage can do a better job at killing whatever is inside that it had eaten and is in the phagolysosome. Okay. And here is another function of a helper T cell that can interact with your B cell by recognizing the antigen, right? And tell the B cell to start producing antibodies and to do some class switching depending on whatever pathogen was found, okay? So we're gonna look into more details now. So here a bit of a step back on the development of T cells, right? So we know that when those T cells are coming from the bone marrow and they are arriving at the thymus, they are not yet committed to T cell lineage. But in the thymus, okay, so this is a part that gets a bit tricky sometimes, so let's wake up, right? In the thymus, you have stromal cells that temporarily produce this factor that is called Notch1, okay, or just Notch, okay? So you have this factor here that is called Notch1. Now, when those cells that are not yet T cells, okay, they arrive there, they get in contact with Notch1, they start to upregulate certain transcription factors, okay, like GATA3, for example, okay? So Notch1 activates expression of GATA3 on the lymphocyte, and when this happens in the thymus, that is when you have a commitment of those primary cells, you have a commitment to becoming thymocytes. Okay, after exposure to Notch3 and transient expression of this transcription factor that is GATA3. Okay, so I put some observations here saying that this expression of this transcription factor is not going to be happening forever. It's only temporarily until you get your T cell to fully develop and be tested and come out of the thymus. By the time your T cell comes out of the thymus, it is not expressing GATA3 anymore, okay? Because by the time your T cell leaves the thymus and goes to secondary tissue, okay, the expression of GATA3 is actually a signal to differentiate the T cell into a subtype of T cell. Okay, so GATA3 happens here when you have commitment to T cell lineage, but then it stops before the cell leaves the thymus, and then later on, if it upregulates GATA3 again, then something else is going to happen. Okay, the most important thing of this slide is commitment to T cell lineage, and that's because of this factor that is Notch1, and who is producing that? That's the stromal cells. Okay, stromal cells in your thymus. Everybody cool? Okay. All right. So now, the cell is going to become a T cell. Okay. Now, remember when you start getting all the T cell receptor recombination, you start with the beta guy, right? Beta thing, and then you go for the alpha thing, right? Okay. Now, there is something else that I didn't mention before. There is an alternative way of T cells to get the receptor. It has nothing to do with the alpha beta. So that alternative way is called the gamma delta T cell receptor. Okay? So this gamma delta T cell receptor, what happens is that, actually it is at the double positive stage. Okay, what happens is that those gamma delta T cells, they're not going to behave like regular alpha beta T cells. Okay, they are going to be able to recognize pathogens in 3D. So those are going to be very, very important for mucosal immunity. Okay, so keep that in the back of your mind now. Okay, and we have seen the rest of the story here. Right, we have seen the rest of the story with the alpha beta, going CD4 or CD8. First is double positive, then single positive, blah, blah, blah. We know that story. Okay. All right. So here, at the double negative stage, it can, instead of become alpha beta, it can go and become gamma delta. We're going to see more on that. All right. So the story with all the T cells that are trying to get a receptor on their surface, okay, a lot of them are trying, and 90% of them are not successful at getting a good receptor. So this is why we have this graph here. 90% of the cells that get to the thymus and really try to get a functioning receptor, they can't do it. So what happens to them? They die. They just left in there, they're not getting the stimulus, they die because they're neglected. And if they're neglected, what's the purpose of life? I'm totally neglected, nobody thinks about me, and then they die. Very sad. Okay. Then you have 20% or less, actually 10%, no, my math, right? If this is 90%, this has to be at the most 10%. And look, 90 to 96%, so this could be 4%, right? So what happens? You have a certain percentage here, okay, that they do have just the right amount of interaction, they have good receptors, they survive, they go to the periphery, and they do their job. And here basically is 2 to 5% of the population. Okay, and then you have the ones here that they have very high interaction with those MHCs, right? So those guys here, two things can happen to them. Either they can die, or they can become T regulatory cells. And the percentage of cells that become Tregs is actually quite small. Okay, and then you have another percentage of cells here that have extremely high interaction with those MHC molecules, and those cells are dangerous. Okay, and we're going to see later why they're so dangerous, but mainly that's a very red flag saying that those cells have to be destroyed, okay, in the thymus. They should never come out. So we mentioned that. All right. One thing here that I had not mentioned before that has to do with your thymic epithelial cells, right? You have a signaling molecule that is called thymus, and is produced by those thymic epithelial cells, and that triggers the positive selection of your thymocytes. So one more term for what is happening with the thymocytes when it's in the thymus. Okay. So here, what's the story with those gamma delta cells? All right, so the cell is in the thymus, decided to become a T cell. It can go through the regular path. Okay, so we got to the thymus here. Blood vessel, thymus-seeding precursor. I got to the thymus right here. It's double negative, right? So still when it's in a double negative stage, it can decide to become a gamma delta cell. Okay, now if it becomes a gamma delta cell, it's going to behave totally differently than your alpha beta cells. Okay, so how is it that your alpha beta cell is behaving? Well, remember, your alpha beta cell is going to start expressing CCR7, right? And then CCR7 is going to allow your alpha beta cell to migrate to the medulla, and then it's going to become single positive, and then it's going to go to the lymph nodes, right? CCR7, that should ring a bell, right? Okay, now those gamma delta cells, they are not going to express CCR7. So once they get the alpha beta receptor, they're just going to come out of the thymus and go straight into the circulation. Okay, so look at this. They don't have CCR7. They're not going to go to the lymph nodes. They are not going through the process of positive or negative selection. So those gamma delta receptors, we have to trust them that they're not going to attack us. Okay. And we really don't have a good explanation of how that happens, because not a lot of research is really viable with these cells. We can't really study it in animal models because animal models don't really have those cells, and they're different cells. So we would have to use humans, and that's not really, easily approved by IRB and things like that. So we kind of stuck with very, very little information about these cells. So what do? They leave the thymus having this gamma delta receptor, and they go to mucosal tissues. Okay. So here, the other type of T cell, right? So this is the gamma delta. Where do they go? They go to the mucosa, they go to the skin, lungs, and uterus. That's where they hang around, right? What do? Well, they're going to behave like natural killer cells. So they're mainly part of your innate immune system, okay? They're going to kill cells, all right? They're going to recognize pathogens in a 3D form, so they don't really depend on MHCs, but they can interact with MHC-like molecules like the CD1, okay? They also can recognize some lipids and some glycolipids, okay? All right, so here, they directly recognize the pathogen, and they can also recognize a stressed cell, okay? And their job is to recognize and kill. So here is a little explanation that we don't have a lot of experimental models. So how do you compare the gamma delta with the alpha beta? So here you look at the alpha beta receptor. So here's a beta chain, here's the alpha chain. That's the site with the hypervariable region where you are going to bind your antigen. And then you look at gamma delta, you have here your delta chain, you have here your alpha chain, and here is a site where they're going to recognize something, so they kind of look the same, okay? Remember, gamma delta is going to see in 3D, and they're going to interact with the other molecules like CD1, and these other two molecules, we haven't seen them before, MIC-A and MIC-B, yes? Yeah. Yeah. So the B cell, the T cells, because of the alpha beta T cell receptor, they have to see things presented stretched out on MHC molecule. So that's 2D stage. So these guys here, they see 3D. Okay, they see things in their regular innate or regular non-denatured state. Okay, now when you get those receptors, either you get alpha beta receptors or you get gamma delta receptors. You're not going to get alpha delta or any of these other combinations. It's either alpha beta or gamma delta. That's it. No mix and match here. Okay. So here is a little chart comparing your alpha beta and gamma delta receptors. Okay. Remember that gamma delta is going to see things presented on receptors like CD1, but you also have those MICA and MICB. Okay. All right. So here, when your cell gets to the double positive stage, all right, so remember gamma delta is coming out of the double negative stage, right? So now let's say your cell, your thymocyte is developing, it's going to be double positive. So now it even has more choices on what to become, okay? It doesn't necessarily have to become CD4 or CD8, right? So look what happens here. At the double positive stage, that cell, that precursor cell, can become a natural killer T cell. And in order to do that, it's going to get a weird T cell receptor that is alpha alpha. All right, that's weird, all right? Because we see alpha beta. This guy is alpha alpha. Okay, that's strange. Now, these natural killer T cells, not only they have a weird receptor, but this receptor here is also not going to interact with MHC molecules. It's going to act more or less like the gamma delta guy. Okay, so it's going to recognize CD1 molecules, and it's going to recognize 3D stuff, right? So it's mainly part of the innate system, okay, not really of the adaptive system. So that's another variation of what can happen to the development of those T cells. Besides that, you can have, from the double positive, you can have differentiation here into a single positive. So the cell can go either CD4 or CD8, right? We have seen that. We totally recognize what's happening here. But instead of becoming helper T cell or cytotoxic T cell, they have also other avenues. If they get a CD8, they could become what you call intraepithelial lymphocyte, IELs. Okay, and those basically also behave like innate immune cells. And they have this job, like the natural killer T cells, they have the job of patrolling the environment under your skin, right? And basically they are quite mean in a sense of if they find something that they think it might be suspicious, they never ask questions, they go there and they kill. If it is something that wasn't, offensive or wasn't harmful, it's like, oops. But they kill. Okay, they kill first and they never ask questions. So they're patrolling, ready to kill whatever they think is odd. So these guys here are CD8 positive and they're patrolling under your skin. Okay. And besides that, you also have an option of those cells, like we mentioned before, that are not going to become helper cells. They're going to become suppressor cells, and those are regulatory T cells. So this is a subset of your helper T cells, okay? But they're really suppressor cells. They're not going to be activating anybody. They're going to be suppressing everybody. So these guys here like to kill everybody. This guy here likes to suppress everybody. Okay. Very positive crew, right? All right. So here we got them, and this is coming out of your double positive stage. All right. So here, when you look at your natural killer T cell, still that's not the whole story. You can have different types of natural killer T cells. They're all working like they're part of innate immune system. You have natural killer T1 or 2 or 17. Could be 3. Well, it's not. It's 17. So your natural killer T1 is very good at producing interferon gamma. Your natural killer T2 is very good at producing interleukin 4 and 13. Okay, so here you have gamma, here you have 4 and 13. And your natural killer 17, thank goodness, produces interleukin 17. At least you can remember that. Well, I guess your immunologist friends here said, well, since this guy is not a natural killer 3, it might as well be named after the cytokine that it produces. Okay? I'm going to see importance of those cytokines. All right. So when you look here at your cells that are patrolling under your skin, okay, they are really part of your innate system. And you have not only those cells that derived from alpha beta, but you also have those cells that are gamma delta. And mainly the jobs of those cells, the job is to figure out who looks suspicious and kill them. All right. So here is a little paper saying, you know, what we know about those cells. So you can just read this, but it's basically what I have mentioned. And again, we cannot really do a lot of experiments because we don't have a good animal model. And look, this was published in 2023, so this is like kind of a recent publication. Oh, finally, first question. Okay. Where are the gamma delta cells normally found? You should take 5 seconds to answer that. I have a divide here. So gamma delta are not natural killer T cells. Right? Gamma delta is coming out of the double negative stage. The natural killer T cell is coming out after the double positive stage. Okay, 10 seconds. What is your answer? E, in your gut mucosa. And several people that said on the skin. No, that's your natural killer T cells that go to the skin mainly. Your gamma delta cells go to mucosal tissues. Okay. So here, this now is when we go to the T cell activation and career path. Yes? Yes, but mainly in the mucosal tissue. So you're telling me you want both answers to be correct? Okay, I'll put both answers correct. Yeah, no, I like to give points. But yeah, but mainly is in mucosal tissue. Yeah, you can find a little bit of them in those other areas, but mainly, yeah, mucosal. And even that way, if you look, okay, what are the choices? No, the choices that you just read. Gut, lungs and uterus, that's all mucosal. So skin is a very small percentage, but mainly you find your natural killer T's there. Okay, a little bit of gamma delta, but mainly natural killer T. Yeah, so I guess it's a bit ambiguous for that answer, but yeah. I will not give such choices in the test, you know. Okay, you have more question here? No? No? Okay. All right, so story with the T cells. It gets really fun. You have stimulatory receptors, you have inhibitory receptor, you have stuff that goes on outside of the cell and is influencing the cellular development. You have stuff that goes inside of the cell, goes on inside and is influencing the development. And then you're going to have different types of immune responses based on the career path that your T cell decides to take. So you're going to have type 1 response or type 2 response. And those you absolutely have to know. Okay, so reiterating here, what is needed for you to have a T cell response? First thing that happens is your innate immune system, mainly your dendritic cells, are going to detect pathogen. It's going to pick up the pathogen, chop up the pathogen, present on MHC, right? MHC class 2, and show up to the T cell. Depending on the pathogen, the dendritic cell is going to start making some cytokines. Now, how does it know? Well, remember those pattern recognition receptors that you have something that look like a little hook, you have T-like receptor, 1, 2, 3, 4, 5, 6, 7, 8, 9. You know what I'm talking about? Toe-like receptors. Does it ring a bell? You have like 4 or 5 for viruses, 9 for DNA that is in bacteria. You have all those different types of receptors that are recognizing different patterns, right? So when the dendritic cell activates a particular receptor, toe-like receptor or pattern recognition receptor, that receptor sends a signal to the inside of the cell for the dendritic cell to express certain cytokines. Okay, now keep that in mind. That's very, very important. Okay, these cytokines that are produced by the dendritic cell that just ate the pathogen, those are called polarizing cytokines. Okay, polarizing cytokines. What are they going to do? They are going to tell the T cell to start doing something and choose a career path. So the polarizing cytokine is going to influence the T cell. The T cell is going to start to upregulate some transcription factors in their genes, and those are called master transcription factors. The T cell is going to decide what to become. After it decides, it's going to start making some cytokines. Those cytokines are called effector cytokines. And yes, I have pictures for that. So here we have a picture, okay? Simple, straight from your book, okay? So what happens here? You have your cell with your pattern recognition receptor. It ate something and it chopped up something. It is producing polarizing cytokines. Okay, so here polarizing cytokines are interacting with your T cell that is naive. Well, now is not naive anymore, right? It has just seen the antigen presented, right? So here you're going to have, because of these interactions, there are at least 2, you're going to have a third interaction here with cytokines. What are the name of those cytokines here produced by your antigen presenting cell? Just to make sure everybody's awake. Polarizing cytokines. And then now you're going to upregulate some master genes. That's the DNA right there. And you're going to make stuff that is your effector cytokine. Now we're going to go into more details of that. So let me see here. This is a bit repetitive. I should have put it up there with the development. I'm not going to go into this because we have seen that before. Okay, we know if one signal becomes zombie, we have seen that before. Okay, so here, the signals that are given to the T cell, we have seen that if you give only one signal, what happens to your T cell? I always appreciate that answer, like nothing. Exactly, right? Nothing happens to your T cell. It becomes allergic, it becomes the zombie. Right? Now you're going to need your second signal. The second signal, we had seen one type of signal that was B7, right? That interacted with CD80. So where is it here? CD28, which one is it? B7 is your CD80, interacts with CD28, right? We have seen that one, and there was a signal for your T cell to do something, to become something. There was a second signal for differentiation. But that's not the whole story, because you have a lot of other receptors on your T cell, and you could have a lot of other signals that are on your antigen presenting cell, okay? So the way your T cell is going to behave depends on what kind of signal is going to interact with what kind of receptor. Some of those signals are going to be stimulating. Some of the signals are going to be inhibiting. So now you have to figure out which one is which and how the cell is going to behave. That's why you have this guy here thinking, oh my goodness, so many options on what to do. Should I become a zombie? No, because I have more than one signal, so I got to do something. Okay? So let's see here one by one what's going to happen. When your second signal is stimulatory, meaning a co-stimulatory receptor, right? So you're going to cause something to happen for the cell to do something, okay? So that co-stimulatory receptor usually is your CD28, right? And you have another one that we have not seen before. It's called ICOS, that is inducible co-stimulator, okay? So those are ICOS or CD28. When those are stimulated, as we have seen before, in case of CD28, they interact with the B7, right? CD28 interacting with B7. You have what is called priming of your T cell. Where is your T cell? This is your T cell right here. Okay, this guy here is your T cell. Okay, so one signal recognizing the antigen plus your B7 CD28, that is called priming of your T cell. Now stuff is going to start to happen. Okay, within 48 hours of priming, your cell that was quiet, meaning it was on G0 stage of cell division, meaning nothing was happening, right? Within 48 hours, this cell is going to come out of the G0 stage and is going to start dividing, okay? And it is going to start to make a couple of things that are important, like interleukin-2. You remember what the interleukin-2 is going to do? It's going to work on the same cell that is auto-regulatory, right? It's going to work on receptors on the same cell. But then remember that receptors in this cell, it had like two parts, but it needed a third part to be really good at binding to interleukin-2. And that part was what? You remember? Alpha chain of the receptor for interleukin-2. So this cell, once it was primed, is going to start dividing, is going to make interleukin-2, and is going to get the signal of interleukin-2 to do some other stuff. So this is all part of the priming. Okay, everybody cool? Okay. Am I going too slowly? Okay, I can speed up. All right, so here. So what is happening here? When you have your stimulus that is co-stimulatory, okay, you're going to have activation. Again, we already saw CD28. Another one is your ICOS, okay? Now, ICOS is really cool because it's not present on a cell that has not seen antigen. Only after you already get some stimulation from CD28 that you are going to have expression of your ICOS receptor, okay? Then your cell that has seen something can do something else, like, for example, become a memory cell, okay? If it didn't see anything, it's not going to remember anything, so it doesn't make sense for this receptor to be activated. But if it has seen something, it can start making the receptor, and then it can become a memory cell. Memory cell is a type of effector cell, but it could also become other types of effector cell. Okay, so here, this interaction of ICOS with the ligand ICOS is very important for formation of memory T cells. So that you have certain inhibitory receptors. And those receptors here are the only two receptors that you have to remember. CTLA4 and PD1. Okay? They are not expressed when the cell is naive. They are not even very much expressed when the cell first sees the antigen. but a very slow production of those receptors start when the cell starts the differentiation into effector cell. Okay, so when the cell is going to choose a career path, it is going to start to upregulate those inhibitory receptors. Okay, if those receptors are upregulated, that means like upregulated at a high frequency or at a high density, that means the cell has been hanging around for quite some time doing its job, and most likely the cell is not needed anymore. So you don't want to have an effector cell that is on for a long time. This is why gradual expression of those inhibitory receptors is really, really important, because those receptors are going to interact with B7. Okay, CTLA4 is going to interact with B7. And you say, wait, but isn't CD28 interacting with B7? Well, yeah. Remember, this guy is stimulatory. So if you have B7 that keeps on interacting with CD28, the cell is going to keep on getting stimulated. Now, if this guy has the same type of concentration or expression at the surface and is close to B7, it is going to steal the interaction with B7, is going to gobble it up before CD28 can interact with that. Okay, that means this thing has higher affinity for B7. If both of them see B7, this thing is going to jump on B7 first. Okay, and that means when CTLA4 gets activated, it is going to put the cell down. It's going to tell the cell to be quiet, to not do anything else. Its job is done. It was hanging around for long enough, and it should just be quiet now and prepare to die. Okay, so those are inhibitory receptors. Okay, we don't have. Now, importance of upregulating those receptors is that it maintains peripheral tolerance, meaning that if any T cell goes crazy and is active for a very long time, you could start having autoimmune diseases. But the production of those receptors are really a checkpoint on not developing those kinds of autoimmune disease. So it's basically promoting tolerance. Okay, so here we have this picture of this CD4 positive T cell saying, you know, oh, but I'm helping, I was activated. And you're like, no, you're going to be quiet now. Okay, I'm helping. So here, co-inhibitory receptor here is your CTLA4, and it is going to compete, okay? It is going to compete for the same type of ligand. Okay. Another inhibitory receptor is this guy here, PD1, and the name of it is programmed cell death 1. So guess what this thing does? It causes the death of the cell that has it, and it's the T cell right there. Okay. Programmed cell death. That things get activated, the cell is going to commit suicide. Okay. All right. So here, stream. So is going to inhibit T cell activation and is going to cause apoptosis of the cell. Okay. Pretend this thing here is not there. It's just the eye of the cell looking at the other one. So it's scary. If the cell doesn't have a nucleus, let's say that this cell is looking red. Let's see what's that. Why am I doing that? Red blood cell. That's the only cell that doesn't have a nucleus, right? And the question is like, why is it that your natural killer cell, when it looks at a red blood cell, and the red blood cell does not have MHC1 on the surface, how is it that the natural killer cell doesn't kill your red blood cell? Well, it's because the red blood cell has inhibitory signals. They're going to tell the natural killer cell not to kill it. Okay? Now, if you have another situation in which you have a cell and you have here an activating molecule, and the cell, okay, the cell doesn't have MHC1, but it has activating molecule. It doesn't have MHC1 because it's infected by a virus, right? So natural killer cell goes in there, and it sees the cell doesn't have MHC1, and it sees activating molecule. Now the cell is going to kill the target cell. Okay? All right, so let's see here. What are the receptors that you should know about? This is it. We have just a few of them here, okay? Mainly 4 of them. I took that from your book. It's a modified version of the table they have because they have tons of receptors and interactions, and I think that's totally overwhelming, right? So what you should know here, you should know your CD28, ICOS, those are stimulatory, and then you should know your inhibitory, CTLA4 and PD1. Okay, you should definitely know what are the functions of those and what they're interacting with. I'm going to ask you about that. Yeah. Okay. All right. So now, focusing on the path for, career path for your helper T cells, right? They can become all different types of things. And remember, your helper T cell is just a regular T cell we have been looking at. Single positive, CD4 positive, alpha beta T cell receptor. So now this guy here is naive. We have never seen antigen, but you know that if it sees the antigen presented by the dendritic cell, it can become all different types of cells. It can become Th1 or Th2 or Th17 or follicular helper cell. Okay. And besides that, there is still a path that it could become a regulatory cell, right? So it has to make a choice, okay, depending on the type of pathogen that the dendritic cell has presented to it. Okay. So how is it that you're going to get Th1 cells? Basically, when you have intracellular infections, you get Th2 by parasites. You get Th17 if you have fungal infection or bacterial infection, but mainly extracellular bacterial infection. Because in case of fungal or bacterial infection, you need a lot of inflammation. And Th17 are very, very good at causing inflammation. Okay. Now, these other guys here, follicular helper cells, they are useful only in the lymph nodes or secondary lymphoid tissues, because they are going to teach B cells how to become memory cells. Okay, they're going to work with the B cells in the lymph nodes. Okay. So let's look here at what is happening, right? How you're getting the stimulation of your T cell. Okay, so your T cell. Okay, let me see. When it gets together with your antigen presenting cell, so where is your T, where is your antigen presenting cell? This guy is your T cell, this guy is your antigen presenting cell, okay? So T cell receptor is here, it's going to interact here with MHC2. Okay, so here's first interaction, and that's going to be second interaction. Now, look what is happening here. At the site where you have these interactions, you call that site, when you have your MHC molecule with antigen interacting with a T cell, you call that site central supramolecular activating complex. You abbreviate that CSMAC. So it's right there. Here you have T cell receptor interacting with MHC molecule. So this area right here, okay, this is also including CD8 and B7 interaction. This area, central area right here, for first and second signal, you call that CSMAC. Now, besides that, you're also going to have areas of cell adhesion molecule interaction very close to this central interaction, and you call this peripheral supramolecular activating complex, or PSMAC. So you have CSMAC, on the sides you have PSMACs. And the reason for that is that you get a better adhesion and better communication of those receptors and ligands, okay, for activation or suppression of the whatever is supposed to do on the T cell. I think I put the video for how the C-SMAC and P-SMAC is working, so make sure you watch that too. Okay. So now, what to become in life? If you're exposed to cytokines that are interleukin-12 and interferon gamma, the Th0 cell is going to become Th1. If it is exposed to interleukin-4, it will become Th2. If it is exposed to interleukin-6, 23, and TGF beta, okay, it will become Th17. Now, from this initial stage of all the cytokines, you should say something here. You should see something here like interferon gamma has to do with fight against viruses, okay? Because interferon is the first line of defense against viruses. So to fight viruses, you're going to need a type of reaction that is going to use Th1 cells because those are very important for intracellular pathogens. Okay. If you look at this, you're going to say interleukin-4. Well, right now we don't really associate anything with interleukin-4, but that has to do with helminth detection and fight. This other guy here, does interleukin-6 remind you of something? Remember the triad for inflammation? What are the 3 important cytokines that promote inflammation? Interleukin-1, interleukin-6, and TNF, alpha tumor necrosis factor. Here, you don't see the others, but you see interleukin-6, and you say, oh, this has something to do with inflammation. Oh, Th17 is the cell to handle inflammation, to cause inflammation, okay? All right, so more details here. Now, different career path. Each one of those is going to produce a certain cytokine, defining cytokine of each type. You're going to go one by one, okay? So Th1 is going to be interferon gamma. What is the most important job of interferon gamma? Is to activate macrophages for them to destroy the thing that they have eaten, okay? When you look at this guy here, Th2 cytokines that are produced, interleukin-4, 5, and 13. Important job of these interleukins, as we're going to see in a few slides down the line, is first of all, interleukin-13 is going to increase the frequencies of peristalsis. What does that mean? Guess what that means? Huh? You poo? No, that's very gentle. You're going to have the diarrhea of your life.
No, I said food.
Food? Oh, no, I thought you said poo. I was expecting to hear something like shit. Sorry. But 13 is a lucky number, increases peristalsis, and you're going to be stuck to the toilet, like for a long time. It's very powerful, okay? So when you think about diarrhea, you should be thinking about some kind of helminth worm infection, because usually those go to your intestines. Okay, so that interleukin is very important at trying to expel the helminths out of your system. It will expel everything else as well, but hopefully the helminths will go with it. Okay, so these guys here, they're going to try to expel the helminth, and they are going to activate cells that can fight those helminths, like eosinophils. They're going to work with degranulation and activation of eosinophils. Okay. All right, so 2 of those. Then you're going to have this guy that is very important for inflammation. Is going to recruit a lot of neutrophils and is going to help you to fight whatever blood infection or inflammation you're going to. So, yes, before you run away. Here, one more question. Which of the following cytokines promote Th0 to Th1 differentiation? Make sure you got your points. Technically speaking, I still have 3 minutes.
Oh.
Anybody need more time? Everybody's done? Okay, what's your answer? D. Somebody switched right in the last minutes. Second. Okay, D is, all right, there we go. D is your answer.